All right, hello everyone, and welcome to the second session of AGSX Virtual Symposium. I see the attendee list is still growing, but we can start here with some announcements. Today's session is all about arthropod genomics. It is the second session in the series, but if you missed the first session, do not fret because they were recorded. Computer's a little slow to respond, but let's see. There's information on the following page. One moment. While I'm waiting on that, I should probably introduce myself. My name is Lindsay Perkin. I'm the organizer of this session, and I'm also going to be the moderator for today. So if anyone has questions throughout the symposium, please don't hesitate to put your thoughts and your questions in the Q&A box down there. All right, I don't know why my slides aren't advancing, but oh, here we go. So I put this link here at the top. This is a very important link. It leads you to all the information. So if you missed the first session on insect genomic technologies to improve food applications, you can follow that link and find the recordings that are on the I5K YouTube channel through there. You can also find more information on the upcoming sessions. So there's two more following this session. There's one on April 13th, application of new genomic tools and techniques in arthropods. That's organized by Dr. Marcel Lorenzen from North Carolina State University. And she has a great lineup of speakers, including herself and Omar Akabari, who is at the University of San Diego, California, and co-founder of Agrigene. And also Clay Chu, who is a research scientist at All Things Bugs. And then the last session is on May 12th, and that's the honeybee workshop. That is organized by Sonia Enyard and Alan Bergnall. They also have a fantastic list of speakers. So if you want to learn more about upcoming sessions and the speakers that will be in those sessions, please follow this link above. Also, if you have colleagues that you think might like this series and have not yet registered, that information is at this link, and also information on the Slack channel where we can have more of a discussion about these wonderful talks today at a later time following the 2 hours that we have blocked out today. So without further ado, we can move into the session for today. So arthropod genomics and genome engineering. And as I put this symposium together, I realized I have a lot of personal bias. So there's actually a better name for this symposium might be arthropod genomics and evolution, because there's a common thread of evolution through all of these three talks. And so I hope we can have a really great discussion at the end about the evolution of arthropod insect genomes and insect life histories. So without any more here, I will stop sharing my screen. We can queue up our first speaker. Our first speaker is a PhD candidate, Amanda Stahlke. She's in the Bioinformatics and Computational Biology program at the University of Idaho, Moscow. And she's going to be talking to us today about the rapid evolution and development of genomic resources for the tamarisk beetle.
My name is Amanda Stahlke, and I am truly excited and honored to present as part of the AGSX Virtual Symposium series. I'll be sharing with you some work from my dissertation on range expansion, admixture, and hybridization in a biocontrol agent, the tamarisk beetle Diorhabda, shown here. So to start, biocontrol, specifically classical biological control, is the introduction of a highly host-specific natural enemy, an agent, from the native range of an invasive species to control that pest or the target in the invaded range. And this is an important tool for resource managers with a cost-benefit ratio of at least 1:8. And from an evolutionary perspective, they're quite interesting because compared to their invasive target, agents are a planned invasion with known source populations, intentional releases at known introduction sites with intentional release numbers, and follow-up monitoring to track those populations. So combined, these systems, biocontrol agents, represent excellent and important systems for the study of novel eco-evolutionary dynamics. Now, to focus in on my focal system, I will take you out to the desert riparian Southwest, to this riparian ecosystem here. Tamarisk is this kind of pale shrub along this riparian corridor. It is thought to occupy at least 360,000 hectares all across the West, shown in these green squares from Eddy maps. As an invasive species, it outcompetes the native flora like cottonwoods and willows, partially by disrupting the mycorrhizal associations in the soil. It overstabilizes the sandy banks with this extensive and deep taproot system. It restricts access to the water for both wildlife species and recreationists. And so it's undesirable and unfortunately very difficult to control. This requires taking out crews to these remote locations with heavy machinery, and an herbicide has to be applied very quickly to prevent resprout. And it's just not practical to control tamarisk in this way for a lot of locations. So this makes it an excellent candidate for biocontrol. And the first agent that was released came from Northwest China, indicated by this star here. This was Diorhabda crenulata, and it was introduced in Wyoming, Colorado, and Nevada with some success. However, these initial introductions from Northwest China failed at southern latitudes because Diorhabda crenulata dropped into the leaf litter too early, leaving it vulnerable to predation and depleting fat storage, while there was all this fleshy, delicious tamarisk still out through the fall and winter. So this was the situation in about 2001, with some control initiating in the north, but tamarisk in the south was still not able to be controlled by biocontrol. Establishment rates improved with additional source collections indicated in these stars across the diverse range of tamarisk. So in addition to Diorhabda crenulata, Diorhabda sublineata, carinata, and elongata were collected from Northern Africa, Tunisia, Greece, and Uzbekistan, and an additional crenulata population from Chilik, Kazakhstan. And between 2006, 2010, these additional source populations successfully established in these more southern latitudes, which is great. But despite no intermediate morphotypes across their native ranges, these Diorhabda species were shown to readily hybridize in the lab. The three, sublineata, elongata, and carinata, did not hybridize readily with crenulata. This hybridization in the lab led us to suspect that hybridization in North America would be likely. And so this is the situation when we started this study. I'll show you these observational presence points for Diorhabda across the Western US. These are data collected by River's Edge West, and each successive year is a different color. So between 2007 and 2014, the points will play here. We have crenulata in the west where it was introduced and constrained by that photoperiodic limit just south of the Colorado-Utah border here, and the hybrid zone in New Mexico and Texas where all 4 species were released. So 15 years after the major releases in 2014, when we were looking at this, we had 3 key questions. That's which releases established and spread, are they hybridizing, and what are the genomic consequences and mechanisms of this rapid range expansion and gene flow among all of these source populations. So for the rest of the talk, I'll present our results. We developed genomic resources to understand evolution in this biocontrol system, first by taking a molecular survey. That's the bulk of the work I'll show. But I'm also excited to present these genomic resources that we're developing, high quality reference genomes. So to get started here, we did assemble a cost-effective assembly of Diorhabda crenulata with flow cytometry. This has a relatively manageable genome size, and so we took a crack at it. The first thing we did was perform shotgun sequencing of 5th generation inbred male. This has low input DNA requirements and is pretty cost effective. And we assembled those breeds, paired N 250, and it actually turned out pretty all right. Lots of contigs, but a relatively high rate of single copy orthologs. I'm just showing the single complete orthologs from the Busco set, and an N50 that is all right with a total length about what we expected. We worked to improve this by generating 10x synthetic linked reads with a chromium library, taking the same Crenulata inbred line, this time from the 12th generation. And again, low input DNA requirements and relatively accessible cost, but the supernova method, assembling reads with just the 10x method, did not work very well. I'm not being totally fair because the supernova method was never meant to be used with these smaller genomes. But where these data are really powerful is in scaffolding the contigs. And so we use this arcs and links method to link the contigs of our shotgun assembly with the 10x data and reduced the number of contigs, bumped up our largest contig and 50 and the single copy orthologs. And so great, we have a nice cost-effective assembly of Diorhabda crenulata. The next thing we needed were representative parental populations of the introduced biocontrol agents. So this project is co-led with Dr. Ellen Batumi, shown here. We got to go out to Greece to collect. So we visited the original source collection sites in this northern Greece area we call mainland Greece, the mainland Greece ecotype, as well as Crete. We collected over 100 individuals at 9 sites in the native Elongata range. It was absolutely horrible. I also got to go out to the native source collection for that Fukang ecotype, thanks to collaborators at the Xinjiang Institute of Ecology and Geography, especially Professor Dao Yuan Zhang, and then PhD student, now Dr. Yanfeng Chen. And we successfully found populations there. So we have the reference genome, Elongata, native range collections, and this Fukang ecotype collection. But because I can't spend my entire PhD traversing the native range of Diorhabda, we had to supplement this with lab culture populations, thanks to Dan Bean and the Palisade Insectary. So we have 30 individuals each from Subliniata and Carinata lab cultures. Okay, so we have our parental reference populations now, and now we can go out and collect at these original release sites and expansion fronts to identify what is going on in the introduced range. So we targeted the sites where single species were released, such as Level Wyoming, 1 Wyoming here. And I'm showing here these transparent points. The colors are the original field release sites of each respective species. Okay, and then the square points are the decernulata range, where we think there's cernulata and they've expanded. And then we have this suspected hybrid zone in the circles, or the filled points here. And each of our collection sites is labeled by this numeric alpha scheme, respective to the state where it was collected. So 1 Wyoming was the first collection in the state of Wyoming. Okay, so in total, we had about 550 individuals from 35 sites and 2 lab cultures. We constructed 9 single digest SBF1 RAD-seq libraries following the methods described in Ali et al. And we use 1.4 thousand SNPs with very low missing data amount. These SNPs are RAD-seq loci aligned to the cernulata reference genome. And for structure analysis, we just used 1 random SNP from each RAD-seq locus. And so now I'll present to you these structure results, the bar plots. And these are the K=4 results, which reflect the species identities representative in our sample. And it's the point in the likelihood curve where it plateaus off, so the delta delta K method. Okay, so we have cernulata in the native range, this Fukang site. We have cernulata in 1 Wyoming here, where only cernulata was released, similarly in 34 Utah, 2 Colorado. And we can make the same assignment for these clusters of carinata, sublineata, and elongata in blue, green, and orange, respectively. Okay, and then all of these cernulata sites are indeed cernulata. And as we travel into this suspected hybrid zone, these sites are oriented north to south. So you can see Kansas and Oklahoma are mostly composed of carinata. And then we get into lots of hybridization. And finally, at the furthest south points in Big Bend National Park, 47, 49 Texas, those are mostly sublineata. Okay, so we can break this down further and identify the source populations that were introduced. So we have the Fukang type from that 46 China, and 34 Utah is the only site where only Chilik was introduced, the Chilik ecotype from Kazakhstan. And then we have in the southeast Colorado area a little bit of a mix, because the Palisade insectary used the individuals that colonized western Colorado and brought them over to that region. So we see a little bit of admixture between the Chilik ecotype and the Fukang ecotype. But the dominant spreader down this Virgin River expansion corridor is the Chilik ecotype, which is interesting. And surprisingly, we found cernulata further south in this suspected hybrid zone than was expected at 28 New Mexico and 18 Texas. Okay, so Chilik is the dominant spreader, and we had surprising Fukang establishment. We can break it down in the same way for the elongata source populations, with the mainland ecotype shown here in dark blue and the Crete ecotype in orange. And what is surprising is that elongata was introduced all over the place over here, but really only 16 Texas has a lot of elongata represented, and most sites are hybrids. Most other elongata ancestry is shown to be a hybrid with other Diorhabda species. So only the Crete ecotype established, and barely. Okay, so this is the scene in 2014. And I would say that in general, establishment, spread, and hybridization were relatively predictable. We expected hybrids where all three were in close contact, and populations largely expanded along these river corridors as expected. But there were some surprises, including that Carinulata further south in the suspected hybrid zone, and a lack of establishment for elongata. But what is most clear is that gene flow was widespread in this area in 2014. Now, there are lots of interesting evolutionary biology questions associated with that, but if I'm talking to a land manager or biocontrol scientist, I think what's most important to us is whether hybridization affects efficacy or risk in the system. So hybridization could increase efficacy by generating hybrid vigor or providing the necessary genetic variation required to adapt to a novel environment. But the risk here is that hybridization could cause sterility and crash populations, or perhaps in a worst case scenario, create transgressive phenotypes, for example, altering the host specificity of these populations. Now, we knew from lab experiments that Carinata and Subliniata hybrids and their respective back crosses had the only change in hybridization compared to lots of other phenotypes. This was the only cross that was shown to be different from their parental types, and they had an increase in fecundity measured by the number of eggs laid in 24 hours. And interestingly, this is the hybrid types that we found most common in our data set. So I'm showing here the Q values from structure, and this is symmetrical as a biparental cross. We can think of the Q value as a hybrid index. And what you should see is that there's lots of back crossing. So this also implies a breakdown or a lack of reproductive barriers. So that is quite interesting. Taking this into a genetic diversity context, we can compare these sites to their native range in terms of nucleotide diversity, the number of private alleles, and the inbreeding statistic FIS. Here, the native range is shown in the dark gray, and then these original release sites are in black, and range expansion sites are shown in lighter gray. And sites with hybrids had an increase in nucleotide diversity. We do see some sign of that population bottleneck with a decrease in private alleles compared to the native range. And if we look at FIS, these sites with hybrids actually have the highest inbreeding statistic, but this is actually a signal of the Walland effect, or population substructure measured within a site, and is probably reflective of population barriers or reproductive barriers between hybrids or recent hybridization. Okay, so that's the hybridization story. What about Carinulata? What are the genomic mechanisms and consequences of range expansion over there? Again, looking at diversity, private alleles, and FIS, it's pretty impressive to see how low the genetic diversity is across the native range and all of these original release sites and expansion front sites, despite definitely a signal of population bottlenecking comparing these introduced range sites to the native range. But no signal of inbreeding, even at the expansion front, where we might expect some of those smaller populations to maybe be inbreeding. Okay, so low genetic diversity is not constraining the rapid evolution and range expansion of Carinulata. So how is this happening? I like to think about this in terms of the response architecture. And we're tugging at this knot in lots of different directions. And I'll just briefly make a shout out here to the work that's ongoing. We're looking at the genetic architecture of ecologically relevant traits, specifically fecundity, dispersal, and diapause, as well as the evolution of thermal sensitivity and evolved plasticity at these range expansion fronts. And this work would not be possible without the phenotyping extraordinaire evolutionary biologist Eliza Clark in Ruth Huffbauer's lab. Okay, so that's the Crenulata story. Lots of ongoing work. There's also lots of ongoing work in this hybrid zone. We return to these sites to monitor a time series. So this was 2014. And then when we return to the sites, Kansas, Oklahoma, this East Texas site, 19 Texas, and some of the southern Pecos River sites, 21, 20, and 18 New Mexico, had all petered out. We couldn't find populations there, but there were populations at these sites between 83 New Mexico and 27 New Mexico, 15 Texas, 16 Texas. And so these population codes correspond, and there were some new sites. So in 2018, what I want you to see is that there were a ton of these Carinata sublineata hybrids, and maybe a cline forming between Carinata and sublineata north to south. It's also striking that this elongata stronghold, 16 Texas here in the orange, largely disappeared in 2018 and was again largely Carinata with trispecific hybrids and even again surprising Crenulata far south. We returned to those sites again in 2019, and so now we have this time series where we can start to measure potentially selective introgression, but it's really complicated. This is a clearly dynamic hybrid zone with highly mobile species moving up and down these river corridors. So this is an area of a lot of work, but I'm really excited to see where it goes. Additionally, Eliza and I have both genotype data now and host preference and performance data for 3 of these populations with differing levels of hybridization. Okay, so I showed some polished work and also some exciting directions that we're actively pursuing, but this is the start of a molecular survey that provides a baseline to monitor these expansion and novel hybrid zones. And now I will share with you these beautiful high quality reference genomes. Okay, so I presented this reference genome back in 2019 at the Arthropod Genomics Symposium in Manhattan, Kansas. And I was so proud of it. And then I saw all of these really amazing high quality assemblies with long reads and low input libraries. Some of them produced and presented by the Ag 100 group. And through the magic of a poster session and conference networking, we agreed to collaborate on these Diorhabda species. And so just comparing the reference genome that we assembled and the one that Dr. Scott Geib and Dr. Shana Sim at the ARS Pacific Basin in Hilo, it's pretty humbling to see this drop in the number of contigs from 16,000 to 130 with lots of large contigs. And these are mostly assembled, or many of them are assembled into chromosomes. So here is the high C data. So this is a contact matrix heat map, and you can see that these contigs can be assembled largely into chromosomes. It's so cool. Also, we have an X and Y chromosome indicated in this red and blue arrow here. And then we can validate these further with rad-seq loci. So these are carinulata samples, sexed carinulata samples aligned to this beautiful reference genome and visualized with rad-seq. And what you can see is there's bias coverage at these contigs for these rad-seq loci. So this is super cool, really impressive to see sex chromosomes. But that's just carinulata, and we know there are so many interesting differences between these species. And just as an example, I'm showing you here the alignment rates of rad-seq loci to the carinulata assembly between the carinulata samples, which have a smaller genome than the other three species that hybridize and have a larger genome. So there's about 25% of rad-seq loci that could be harboring lots of interesting biologically relevant data and information about these species. And what is super cool is that we have assemblies ongoing for the other three as well. And carinata so far is the only one that has Hi-C data also integrated. And so we have 100 contigs largely assembled into chromosomes as well. And we'll work in Hi-C data for lineata and elongata as well. Okay, and just to wrap up, I want to show the mapping of Busco genes between the carinulata contigs and carinata, just as a teaser for the comparative work that's coming down the pipes. And here's those X chromosome contigs again, and they have Busco genes that map to what looks like an X chromosome in carinata, and this is all very cool, and I'm really excited to unpack these reproductive barriers, diapause regulation, and more in these genomes. I also want to show you, just for comparison, this is the TCAS 2.0 tribolium from NCBI, nicely assembled into linkage groups. It is quite different from Diorhabda, and so what you're supposed to get from this spaghetti mess is that carinulata presents a new and different chrysomelid reference for all of us. And that's, there is some X chromosome homology. So yeah, I'm really excited for the comparative work across contemporary populations, as well as the deeper historical scales. And these high quality reference genomes can empower lots of inference. I'm very excited. And with that, I have a million wonderful collaborators to acknowledge here, including my advisor, Paul Hohenlohe at the University of Idaho, and my co-lead author on much of this work, Ellen Batumi, and Scott and Jenna at the Pacific Basin office. And with that, I'm excited for your questions. You can find me on the Arthropod Genomics Slack channel, GitHub, Twitter for better or worse, and email. And that is all.
All right, thank you, Amanda. That was an excellent presentation. We have a couple questions that I thought I could at least get started on while we transition to the next speaker. Most of them have to do with your SNP analysis. The first one is asking, for your structure analysis, did you use all SNPs available, or did you perform some sort of filtering before you did your analyses?
Yep, that's a good and important question, and I totally glazed over all of the bioinformatic details there. So I responded in the chat here. First of all, we removed individuals that had low coverage going into the analysis because that generated a lot of missing data. And then after genotype calling, remove SNPs that were missing in 50% of the individuals. And then the structure analysis was only run on a single locus or single SNP from each rad locus. And that code is available on GitHub if anybody is interested or has suggestions on that.
Okay, thank you. And then related to that, someone else writes, given one SNP taken per rad-seq locus and used for the structure analysis, was there any concern or determination of linkage could have biased results in any way?
Yeah, so the idea with just taking a single SNP is to reduce the effect of linkage, but with that really fragmented assembly, we had a hard time testing for linkage explicitly. So yeah, linkage is a real concern, but hopefully by just taking that single SNP with relatively dispersed rad loci, we're mitigating that as much as possible.
Okay. Okay, great. Well, and to keep kind of on time here, I'll go ahead and introduce our other speaker, but we can get to the other questions for Amanda during the discussion after all three of the presentations. I also wanted to remind participants, please don't be shy. Put all your questions into the chat. I think having a good discussion after these talks is contingent on a lot of questions. So please put in questions as you have them. Okay, so our next speaker today is Heath Blackmon. He is an assistant professor in the biology department at Texas A&M University. Heath's lab studies the evolution of chromosome number and sex chromosomes, among other topics. And today he's going to be telling us about models of chromosome evolution.
Okay, I'm Heath Blackmon. I'm an assistant professor in biology at Texas A&M University. And I want to thank the organizing committee for giving me the chance to give this presentation today. I'm going to talk to you about some comparative phylogenetic approaches that we've been using in the lab that incorporate a realistic model of chromosome and karyotype evolution to understand and detect the presence of sexual antagonism across genomes. I think one of the most fascinating things when we look across the tree of life is we look at these species that have males and females, and we see strikingly different phenotypes between males and females. And this implies that there's a very different way of being fit if you're a male versus being fit if you're a female. And this difference in phenotypes that are high fitness, and this is one of my favorite examples here. This is a beetle where the male and female of the species were actually described as separate species until we started having genetic data, and we realized that it was actually two sexes of the same species. This huge difference in what works well for males and females can lead to sexual antagonism, where what is fit for one sex is different from what's fit for another sex. And today we're going to be talking about intralocus sexual antagonism. So this is when there's a single locus in the genome that has this kind of a fitness relationship that I've plotted here, where we have one genotype, say for instance, the A1A1 genotype, that provides high fitness in males, but low fitness in females. And then we have a heterozygote that is intermediate in fitness in both sexes. And then we have the A2A2 genotype that's highly fit in females, but very unfit in males. And this kind of sexual antagonism is a key assumption that underlies our models for how sex chromosomes evolve. And unfortunately, we actually have very little data about what is the frequency of sexual antagonism kind of across the tree of life. Really, the only good empirical evidence that we have comes from Drosophila. And so in my lab, we're wanting to try and understand how common are genes that have this type of fitness relationship between the sexes. And so one way that we might test this is kind of looking across the tree of life for a signature of results that would be produced by having this kind of variation. And we can actually go back to the 1980s. There was this paper published by the Charlesworths that stated that fusions between autosomes and sex chromosomes should be favored, should be more common than we would expect by chance if this sexual antagonism exists. And so briefly, their model, the way that it worked was you could imagine a species where you had a pair of autosomes, and in red here, I'm showing you a sexually antagonistic locus. So that allele with the M on it, that's going to provide high fitness in males, but the allele with the F on it right here is going to provide high fitness for females. When a male that has a genome like this produces sperm, they're going to produce 4 haplotypes. They're going to produce X-bearing sperm that have the female benefit allele and X-bearing sperm with the male benefit allele. And then they'll produce Y-bearing sperm with either the female or the male benefit allele. The problem is that 2 of these haplotypes are going to produce offspring where the sexually antagonistic allele that it carries doesn't match the sex of the offspring that's going to be produced. And so what the Charlesworths pointed out was that if you had a fusion between the autosomes and the sex chromosomes, then you create linkage between the sexually antagonistic locus and the sex-determining locus, such that when this individual makes gametes, they're going to produce gametes that lead to offspring that have a match between the allele that is impacting sex determination and the allele that's impacting the fitness of that sex. And so you can see in this case, you would produce primarily X-bearing sperm that carry the female benefit allele and Y-bearing sperm that carry the male benefit allele. Now, this was published 40 years ago, but nobody has found a way until very recently to provide any kind of quantitative test of this. And so we started thinking about ways that we could test this idea and see, are there more of these fusions between autosomes and sex chromosomes than we would expect by chance? And so the first thing that we did was we just started thinking about what should the karyotypes look like across a phylogeny if these things are favored. And one thing that we realized was that, you know, if you imagine a phylogeny like this with 4 species on it, we imagine some basal ancestral karyotype that we have, in this case, 10 autosomes and an XO sex chromosome system. And we start allowing for fusions between autosomes, these sex chromosome autosome fusions that we think should be favored, and also fissions of chromosomes. We can imagine just laying these different types of mutations down on this tree. So here we have an autosome fission, no impact on sex chromosome system. Here we have an autosome fusion, again, no impact on sex chromosome system. And then here is one of these fusions that we're interested in here, where we're fusing an autosome with the X chromosome. And so we have a transition from an XXXO sex chromosome system to an XXXY sex chromosome system. And with that, we also have that reduction in autosome number. And then just throwing down one more fission here for good measure. And if you think about this process occurring across a large phylogeny, we might expect that if these fusions between sex chromosomes and autosomes are important in kind of shaping the number of autosomes in the genome, that we might be able to compare in a closely related clade XO species with XY species and look for a reduction in autosome number in these species and just see how common is that across a phylogeny. And here I'm showing you with XO and XY, but this same process would happen in, say, a species where ancestrally they had an XY sex determination system, and then we had neo-XYs. This neo-XY is what cytologists will use to describe a karyotype when they see that there's been a fusion of an autosome and a large expansion in the size of the sex chromosomes. And again, what we would see here is that the mean in these XY species is higher than the mean in these neo-XY species. And so we tried doing this. We first went to Polyneoptera. So this is a group that includes roaches, Dermaptera, grasshoppers, mantids. And for every genus in this group that had species both with an XO and an XY, or an XY and a multi-XY system, we asked what's the mean autosome number in the species that have one or the other type of sex chromosome system. And we can look for that reduction in autosome number that's associated with these differences in sex chromosome system. And this is what the data ends up looking like. And so each line in this plot is connecting the mean of the species in a genus with XO versus the mean of species in a genus with XY, sex chromosome systems. And the same thing for XY to multi-XY. And what we see is that almost all of these lines have a downward slope. So we have a reduction in autosome number along with these transitions. And so actually, when we look across all of these genera, we actually see 16 out of 17 genera that have both XO and XY have a reduction in autosome number in the XY species. And we see a less extreme picture when we look at XY and multi-XY. And so those red lines that are going up, that could indicate that there just have been several fissions. It also could be that that multi-XY system isn't coming from an autosome sex chromosome fusion. But overall, the picture that we see here is that it looks like there are a lot of these fusions occurring between sex chromosomes and autosomes. Not all clades show this strong of a picture. We've also done the same analysis across 142 genera of Coleoptera. And here what we see is actually a pretty good mix between these ones that show this clear signal of an autosome sex chromosome fusion and ones that don't. There are still, over 60 genera that have this reduction in autosome number along with the transition. But obviously something else is also having a big impact on chromosome number. And neither this display or this, though, can really capture whether or not we see more of these fusions than we would expect by chance. This data can only tell us that a lot of these things happen. And so we really wanted a way to answer this question of whether or not there are more of this type of fusion, the sex chromosome autosome fusion, than we would expect by chance. And I think the reason that this hasn't been addressed in a robust quantitative framework yet is because it's actually, there's some difficulties involved in addressing this across a phylogeny. And the first difficulty that we encounter is that the probability, how often these fusions should occur, depends on the karyotype that's present in any single part of the phylogeny. You can imagine if you have a genome that's made up of 5 pairs of autosomes in an XY sex chromosome system, 33% of all the random fusions that could occur would join a sex chromosome and autosome. But if we imagine a different genome, a genome with 10 pairs of autosomes and an XY sex chromosome system, well, then only 18% of the fusions should actually join sex chromosomes and autosomes. And so what we really need is a way to calculate this percent that we expect for any hypothetical karyotype, any sex chromosome system, and any number of autosomes that's present. And so that's what we set about doing. And this is an equation that allows us to do this. I know this is a lot of letters, but it's actually a pretty simple equation. If you imagine, so the value that we're getting here is the proportion of sex chromosome autosome fusions. Out of all the fusions that occur, what proportion should be of this type? And so the way that we do this is we start out with this 1 represents all possible fusions, and then we subtract from that the fusions that are coming in from females. In these two terms, this captures autosome-autosome fusions and X chromosome fusions. So these are the only kinds of fusions that can occur in a female that don't involve the fusion of a sex chromosome and an autosome. And so we take that out of this 1. And then we also remove all of the non-sex chromosome autosome fusions that could occur in males. So here we have autosome-autosome fusions, here we have XX fusions, and here we have YY fusions. And so when we do this, what we're left with then is this proportion of sex chromosome autosome fusions. We can, here I'm showing this with X's and Y's. This works equally well with ZW systems or even UV sex chromosome systems. And what it allows us to do then is for any autosome count and for any sex chromosome system, we can calculate this exact value. So this is kind of key to being able to test this hypothesis. And so let's look at how we apply it. The first step in being able to apply this is we actually need to be able to reconstruct the evolution of the karyotype across the phylogeny. And what I'm showing you here is a Markov model that we use to do this. You can see these 2 large external squares. These are our 2 sex chromosome systems that we have on a phylogeny. In this quick case, it's XY and neo-XY. And while we have this XY sex chromosome system, we have 2 possible transitions that can occur. A lineage could start out with I chromosomes, and it could have a fusion event that would lead to I-1. So that would be a fusion of an autosome and an autosome. It could also have a fission event leading to I+1 chromosomes, and so that would be a fission of an autosome. But what's really important here is we also have this arrow that comes from the XY box to the neo-XY box. And so we can have this transition, and this is the kind of fusion that we're interested in. You can see here we're reducing autosome number by 1, so we're going to I-1, and we're transitioning the sex chromosome system. Once we have this neo-XY system, we can still have fusions and fissions of autosomes in that state as well. And so we can take a phylogeny and the karyotypes that we observe in extant species, and we can use this model to reconstruct the evolutionary history of a clade, producing something like this, where we can paint each of the branches in the phylogeny to show what they were in the past. And this becomes really powerful in testing this idea, because what we can do is we can create a table like this where we have each of the different karyotypes, and then we can just look at this tree and count what proportion of the tree is in each one of these karyotype states. We can use this data in conjunction with our equation that we came up with and actually calculate the probability of sex chromosome autosome fusions for each one of these karyotypes, and then weight those probabilities or proportions by the proportion of the tree that's in that state. This then gives us this weighted sum, and what this captures is, across this whole phylogeny that we're looking at, what proportion of all the fusions that happen should have been between a sex chromosome and an autosome. And this is assuming that all fusions happen with equal likelihood. And so what we're going to be looking for is whether or not we see an excess. If these sex chromosome autosome fusions are favored, they should occur at a higher frequency than this expectation under our null model. So once we had developed this process of doing this, we then went to test it empirically, and we went to Drosophila. And so we applied the Markov model that I showed you, and we used a large phylogeny that include both of the major suborders of Drosophila, and we can reconstruct the evolution of karyotypes across this phylogeny, where we have anywhere from 3 to 6 autosomes, and we have both an XY and a neo-XY sex chromosome system. And then we can just count up the proportion of the tree that's spent in each one of these states. And we can use those proportions, just like I showed you in that toy example, to actually generate an expectation for Drosophila. In Drosophila, what proportion of fusions should be sex chromosome autosome fusions? So that's great. That gives us our expectation, our null expectation. We then go back to this reconstruction, and we can actually count how many sex chromosome autosome fusions have occurred, how many autosome fusions have occurred. And so here in red, I'm showing you the sex chromosome autosome fusions. So here we have a transition from blue to yellow. And so we're going from 6XY to 5 neo-XY. So this is one of these sex chromosome autosome fusions. We can also count all of the autosome fusions. That's these black circles. And so here we have one going from blue to red. So actually, let's say red to black. And so that's going from 4 to 3 with no change in the sex chromosome system. And so that allows us to calculate this actual proportion that we observe in this clade. And we can generate a distribution. Now, both of these are distributions, not single values, because there's uncertainty in this reconstruction. So we actually don't use just a single reconstruction. We do this in a Bayesian framework, and we actually generate thousands of possible evolutionary histories. And for each one of those evolutionary histories, we produce one expectation and one inference. And then we can compare these and ask whether or not they match up with our expectation. I hope that maybe a couple of you noticed that this is actually not what we would predict based on that Charlesworth paper that I showed you at the beginning. The expectation there was that these should be favored. These sex chromosome autosome fusions should be favored, which would predict that we would see more of these than we would predict by chance. And so this was a bit mystifying when we saw this, and we had to think long and hard about why we wouldn't see more of these. This actually implies the opposite, that there are fewer of these sex chromosome autosome fusions than we would expect by chance. And so earlier this year, I guess it's last year now, we had to end this paper with some explanation for this result that we saw in Drosophila. And what we said and what we think is going on here is that Drosophila are weird, and that actually these fusions, even if they do have a beneficial effect initially in resolving that sexual antagonism and allowing males to make gametes that are more fit, that because of the genes that are present on the new Y chromosome that's formed, deleterious mutations may reduce rather quickly the actual fitness benefit that is realized by individuals that carry one of these fusions. And let me explain to you kind of why I think this is happening in Drosophila in particular. So we start out with a genome like this. We have our fusion event. And now we have this one large X chromosome and these two Y chromosomes. And the thing is that in Drosophila, autosomes are huge. Actually, the largest autosome in Drosophila is chromosome 3, and it contains fully 43% of all the genes in the genome. For comparison purposes, the human genome, our largest chromosome only contains 10% of our genes. And so that means that this Y chromosome is a huge target for new deleterious mutations that are coming in. And additionally, Drosophila have what's called achiasmatic male meiosis. So that whole Y chromosome that's formed with this fusion is immediately non-recombining. It no longer has a partner to recombine with. And so it's going to be subject to a force called Muller's ratchet that leads to the accumulation of deleterious mutations on non-recombining genomes. And so what I think is going on here, you can imagine starting out with a population where you have this initial fusion that occurs, and it should be more fit due to the presence of sexual antagonism. And so we would expect it to increase in frequency. But this takes time, many generations to increase in frequency. And every generation that goes by, there are also deleterious mutations that are coming into the population. And so I'm showing that with these yellow dots. If this happens on an X chromosome or on an autosome, those are going to still be recombining in females. And so they can be recombined out and you can maintain a high fitness chromosome. But when they occur on a Y chromosome, like this one that I'm showing you here, there's no recombination, so they can't be gotten rid of. And in a finite population, the expectation is that those will accumulate then. And so here I'm showing you after several generations, even the best Y chromosome in the population, this fused version, has 3 deleterious mutations. And so I think what's going on in Drosophila, and the reason that we don't see these more frequently, is that even if you get this initial fitness benefit, before it can reach fixation, it accumulates so many deleterious mutations that it's actually less fit than the unfused version. And so then these are lost from the population and you never see them go to fixation. We've actually already started trying to test this from a theoretical standpoint. So we've built forward time population genetic simulations where we have achiasmatic male meiosis, and we're able to introduce fusions between an autosome and the sex chromosomes. And we can fuse either a large autosome or a small autosome and then look at the outcome of the simulation. And so here's an example of what we see when we fuse a small autosome to the sex chromosomes. This is an autosome that contains 5% of the genes. And what we see is that about half the time we get fixation, a little less than half the time we get fixation of this fusion. But when we change that and we try fusing a large autosome to the sex chromosomes, what we see is that they increase in frequency initially, but then they drop back down. And so when we look at these, what we see when we dig into these simulations and look at the genomes that are being simulated, what we find is that they've fixed these deleterious mutations after the fusion event, which then counterbalances the fitness benefit that they would have due to the fusion. And so I think this explains why Drosophila don't show the pattern that we predicted. Reassuringly, we've started testing this in other clades. We're kind of working our way across the tree of life to understand how frequently this process seems to be playing a role in genome evolution. And when we look in Orthoptera, actually we see this beautiful picture where we actually do have significantly more of these sex chromosome autosome fusions than we would predict. And right now we're working on applying the same approach in dragonflies, Lepidoptera, mammals, kind of across the tree of life to really let us understand how often do we see an excess of these sex chromosome autosome fusions. This is really important because sexual antagonism is this key component of some of our foundational theory for how sex chromosomes arise, how they evolve, why we end up with highly diverged sex chromosomes like humans have. And yet we have almost no empirical support beyond Drosophila for for the presence of sexual antagonism. And so this approach offers a promise of a way to really basically survey across the tree of life and detect the frequency of sexual antagonism among clades. And so take-home message here, I would just say, first off, these fusions between sex chromosomes and autosomes, they're super, super common. Every clade that we look at shows lots of genera that have this pattern where closely related species have this transition in sex chromosome system and then a huge reduction, and then a reduction in autosome number that goes right along with that. We developed this new model and this equation that allows us to test for whether or not those fusions that we see are actually more than we expect. And what we found is that it depends on what clade we're looking at. Drosophila definitely don't show that pattern, but orthopterans definitely do show that pattern. And I think through these simulation studies that we're seeing that the reason that we don't see this in some clades is due to the peculiarities of those genomes and that genetic system, the lack of recombination in males. And so that's going to be another thing that we're trying to tease apart, is the impact of achiasmatic meiosis on our expectation for these results. And couldn't do this without lots of help from people in the lab and funding from NIH and NSF and Texas A&M. And I'll be happy to address any questions that anybody has. Thank you.
All right, thank you so much, Heath. That was a fascinating talk. We have several questions coming in. I think we can address a couple now and then save the rest for the discussion. Let's see. One I was also curious about, what about ZZWZ systems? Do you observe the same trends? Do you see more fusions in groups with holokinetic chromosomes?
So we're just starting to apply this to ZW systems with Lepidoptera, and I don't have any results finished for that yet, but we hope to see it soon. Was the other question you said about holokinetic species?
That was part of that. Do you see more fusions in groups with holokinetic chromosomes?
So I haven't looked at sex chromosome autosome fusions in this, but we actually just published a paper a couple of months ago looking at fusions and fissions across, I think, 13 insect orders that captured 3 clades that have holokinetic chromosomes versus monocentric chromosomes. And there, actually, we don't see a significant difference in the rates of fusions and fissions among those orders. We see far more variation among the holocentric clades, but not a consistent pattern of higher values.
Okay. Okay, great. We have several questions. Did you have one that you found particularly interesting you would like to address real quick? Heath?
Sure. So let me see.
There's actually one maybe short one that might be good. Does your model incorporate fissions?
Yes, so it has fissions. We can expand that. You know, I showed 2 boxes where we have like XO and XY, but we also have an expanded model of that where we also have multiple sex chromosome systems, which I kind of mentioned in passing during the talk, but you can have like an XXY, or in some beetles, you'll have 4 or 5 Y chromosomes. And that could occur through repeated rounds of fusion events, or it could come through like a fissioning of the existing Y chromosome or the existing X chromosome. And so we can add that into the model as well, and actually test which one of those are more likely to explain the presence of these multiple sex chromosome systems.
Okay. Okay, great. Thank you. Okay, everybody, we've been at this for an hour. So if anyone wants to go take a break, get coffee, stand up, shake around, run around in a circle for a minute. We still have one really excellent speaker left. And then following that, we'll have a small discussion. Let's take maybe 1 minute to stretch your arms a little bit while we queue up the next speaker. And I also want to remind everyone to keep those questions coming in the Q&A. And then anything we don't get to during our session right now, we can pick up on our Slack channel. So the link I showed you at the beginning to our webpage, you can find information there in the registration section to sign up for our Slack group where we can have further discussion. So I encourage everyone to do that as well. All right, so last but not least, our final speaker today is Josh Benoit. He's an assistant professor in the biological sciences department at the University of Cincinnati, and his lab focuses on insect physiology. Today, he will be discussing the sex and reproduction in several insects using multiomics approaches.
Hi, I would like to thank the organizing committee for inviting me to give this presentation on using multiomics approaches to examine reproductive biology and kind of unique insect systems. And so I'll talk, rather than talking about one kind of big story, I'll talk about 3 smaller stories of projects that the lab's been working on. So let's go ahead and get started. All right, so mainly this is going to kind of focus on a study on fruit flies, a study on Antarctic midges, and then some of my work over the last few years on insect viviparous systems. So this is the current active areas where research is going on within my lab. We work on kind of multiple projects and that sort of thing, but today I'm going to talk a little bit about kind of mother-offspring conflicts in viviparous reproduction, evolution of sexual ornaments of Drosophila, and reproductive mechanisms of Antarctic arthropods. And those will be the 3 projects I'm going to talk about today. So the first thing I'll talk a little bit about is some of my recent work in collaboration with Mike Pollock. This was mainly done by Francis Tyler, Jorge Orduro Gonzalez, and Cassie Hooker. And this is looking at kind of sex combs and how selection on these precopulatory mechanisms can impact fertility aspects. So what mainly Jorge and Francis did is they actually did selection studies where they could either make the combs bigger or smaller, and then we want to see how this impacted potentially mating, fertility, in those aspects. So just to emphasize how important this is to Drosophila biology, there is a lot of variation in these from species to species. Some have kind of medium size, some have very small sex combs, some have no sex combs, some have larger sex combs, and some have these massive ones. So there's a wide variety of these that occur in fruit fly species in nature. And so the first thing we actually saw is that the ones with larger sex combs denoted by high have a higher success in competitive fertilization. So that was really interesting to us. And so it kind of assumed bigger combs, more competitive fertilization. So we did an RNA-seq analysis to actually figure out what was going on with this. And the main thing that came up in all the ones selected for the sex combs, which are a pre-copulatory aspect, we see increased levels of post-copulatory factors, which are accessory glands, so in the hyaline. So this suggested that maybe something post-copulatory was going on as well. And so we wanted to kind of delve a little bit more into this. And so to do so, we actually wanted to use some laser removal of the sex combs. And so this is Carl here. He's a former grad student of Mike's using this laser system. And what you can do is you can take the ones that have the high comb, and then here's one that's low comb, and you can blast off the sex combs with a laser and then make them small sex comb flies. And so this doesn't take any genetic manipulation or those aspects, but then you can test, all right, what is the actual impact of these sex combs? And so what we were able to see is that we were able to remove the sex combs, make them about the same size as the ones with the low sex combs. And when you actually break these down, you do not see a reduction in the improved mating or fertility. This suggests that there's probably indirect post-copulatory sexual selection is likely to contribute to ornament diversification. So there's potential mechanisms for these linkages, and they're all discussed within this current biology paper that was actually just published about a month ago. So if you want any more details on these studies, feel free to review the paper or send me an email. So the next study I want to talk about is some of our recent work on midge reproduction. So I have been interested in the Antarctic midge since I was lucky enough to go to Antarctica and work on these as a graduate student. And these are actually the largest land animal in the world, in Antarctica. And so, but they're not very big. And so they're endemic. They have a 2-year developmental period, mostly as larvae. They only last 2 weeks as an adult. They're freeze tolerant, but only as larvae, and they lack wings. So they're pretty unique among insect systems. And so the genome has been sequenced for these insects here, where you actually, it was sequenced a few years ago by the Denlinger Lab along with Joanna Kelly. And what they were actually able to find is one of the smallest genomes for insects. So little is known about the actual mechanisms of reproduction in this system. So what we want to do is begin to study this here. And so here is a male midge and a female midge. And so what do they have? So pretty much the males will live longer than the females unless they mate a lot. And that's about it. Females also will lay one big egg mass covered in this gelatinous material here. And so this was some work by Shingoto. And so for our studies, what we want to say is, all right, what are the molecular mechanisms of Belgica reproduction? And so this was a combined study mainly by Jeff Finch, Ben Davies, Carly Preda, and Sonia Nandial, and was published last year in Scientific Reports. The main goal of this was to actually look and see what is this gel and what is its role in Antarctic midge reproduction. So here are some images we have this system here. And so on the left, what you have is the eggs here, and then you have the accessory gland full of this gel. On the right, this is what it looks like following egg deposition. And so you'll see the spit accessory gland and then the spit ovaries. So to actually analyze this, we took the females, males, female accessory glands, male accessory glands, and then we did an RNA-seq analysis of these different organs and then compared them. And so we were able to find what are the genes associated with the female accessory gland, the male accessory gland, and other factors associated with just general reproductive biology in this system. And so this kind of allowed us to actually establish what are the genes in the female accessory gland and the male accessory gland. So, but mainly for the sake of this talk, I'll talk about the gel produced by the females. And so in order to actually confirm what is in the gel, we did proteomic analysis of the gel that was actually secreted by the females. And so we were able to identify the components within this. And mainly it's one uncharacterized unknown protein, a couple of vitellogenin-like ones, a larval serum protein, and that composed most of the kind of overall composition of this gel. The really interesting part, though, is very few of these proteins, particularly some of the ones that are most highly present within this gel, are not the female accessory gland. They're either actually produced within the larva or they're produced within the females. And so very few of them are actually made by the female accessory gland. So this seems to be more just a storage for it, but some of them are made in the larval stage and some of them may be made within the other locations within the female and transported to the female accessory gland for excretion. So what is the role of this gel? So the main thing that we thought is, all right, it's nutrition. So we wanted to, would it provide all the amino acids for early development? And then would it have a glycosylation and phosphorylation to provide those nutrients as well within the genes? And yes, indeed it does. It will provide a complete, all the amino acids, even the essential ones, and then it will be glycosylated and phosphorylated. So it likely provides phosphate and then sugars and then a complete amino acid profile necessary for the developing larva. So we followed that up with some more functional studies where we actually took some of the egg masses and then took some of the little larvae and took them out and either put them on food that they would normally eat that we've established in previous studies with and without the gel. And so when you actually give them the ones without the gel, less of them actually survive, and then they actually grow much slower. So it indicates that likely having access to the gel more than likely to actually feed on is critical to the development of these little larvae. So kind of move on beyond some of the roles of this is we want to see, all right, this little gel is directly attached to kind of surfaces and those. Does it actually assist in thermal buffering of the eggs? So we wanted to see, all right, you'll see these daily thermal changes that occur in some of these areas where these midges live. It can be pretty drastic. And then what we did, we actually took eggs in the gel, eggs without the gel, and then eggs without the gel at a constant temperature. And we see that if you remove the gel, you see a reduction in egg viability if it's exposed to thermal stress. And you'll actually will see as well when you have the ones either within the gel or outside of the gel. So the outside the gel is in the blue up here, and then within the gel is in the orange here. It actually changes the maximum thermal shift in the rates of temperature change. So this gel actually buffers the eggs from thermal changes. So along with actually providing nutrients, it also helps to buffer thermal changes. We also, within this paper, there's some data as well to show that it's very critical to actually preventing dehydration as well. But that's, we kind of assumed that from the get-go. And so I'm just not going to talk about that here. So this is the overall kind of summarize this full project we have here. So mainly, we're really understanding, we really wanted to understand how reproduction occurs in the system. So in the paper, we characterized both male and female reproductive biology, but we were really interested, and I talked about here, of what is going on with this gel, what it provides. And so it provides, it's made within the larva, within the female organs other than the female accessory gland, and within the female accessory gland. It's critical to multiple, it prevents egg dehydration, functions as a thermal buffer, and it acts as a nutrient resource for the developing larva. So there's a few different things that this actually does in relation to ensuring the viability of these eggs developing. So now I'm going to move on to the final of my 3 topics here. Remember, the previous 2 topics, if you need more about those, just shoot me an email. I'll send you the paper if you don't have access, or you can find the paper online. So a big part of my research over the past few years has been looking at live birth in insect systems. And I'll talk a little bit about my work in Glossina and some of my recent work in Diorhabda, a cockroach. And there's been actually a lot of independent origins of viviparity in vertebrate systems. But when you start moving into invertebrate systems, there is has evolved probably potentially at least 400 to 600 times. So there's a lot of evolution of live birth that occurs. And it has a varying different levels. So in some of them, it's only the larva's the individual larva rather than eggs with no nutrients provided, all the way up to things like Glossina, where all nutrients for larval development are provided by the moon. So there's a wide variety of these systems. So again, why does live birth evolve? It's mainly due to the things. It probably protects the progeny from stress, protects them from like varying levels, finding their own nutrients, being restricted to a certain area. And then there's just a wide variety of aspects. But really, I'm going to focus on talking about some of the mechanisms in Glossina and then in Diorhabda here. So this is from an annual review article that Jeff Fitardo, myself, Veronica Mikhalkova, and Srop Ascoy wrote. And this focuses on the reproductive system of Glossina. So they develop a single intrauterine larva that is fed milk from this milk gland. And so this is the process that occurs during birth. This massive larva is actually deposited. So sometimes it weighs as much as its mother. And so a lot of my postdoctoral work and some of my work as junior faculty was actually characterizing the system. So we were actually able to characterize within Glossina morsitans all of the milk proteins. They're kind of like, some of them are like lipocalin, some of them like other milk proteins, very similar to in mammalian systems. Then what happens in this process, and then also what happens when this process stops. There's a rapid change that occurs when this stops to go into the next pregnancy cycle. Both of these are very important for tsetse flies to reach maximum reproductive output. So we moved on from some of these previous studies and actually published this study, it was either last year or the year before, on comparing the multiple Glossina species. And this was a lot of work from Veronica Michalkova, Jeff Attardo, and then my former postdoc mentor, Srap Askoy. And so what we did for this is we actually looked at, all right, what are the genes involved that are enriched in the larva, enriched in the after pregnancy? And then we actually found that it's pretty much almost identical among all these different Glossina species. There's very few difference. They're under very strong purifying selection where there's almost no change at all. They're all highly expressed during lactation. And so the only really major difference is there's actually a duplication in one from one lineage between some of the Glossina species. But other than that, they're producing almost the same exact milk proteins. They're really not changed at all. So this indicates their importance to the reproductive output of this fly. So next I wanted to move into kind of my own studies in this. And so I moved into looking at matchotrophic viviparity in the beetle mimic cockroach Diploptera punctata. They actually have a brood sac that kind of serves as a pseudo uterus and placenta. And then they have a gestation time that's much longer than Glossina. Glossina, it's only about 10 days. In Diploptera, it's about 70 days. And here's some images of what it looks like here. And so this was made pretty much a lot of work by one of my former graduate students, Emily Jennings. She's now a researcher at AstraZeneca, and a very good undergraduate, Matthew Korhauer. And so to understand this system, we didn't really know much about the molecular mechanisms of live birth in Diploptera. So we did microbiome characterization of it, RNA-seq characterization of it, and then also sequenced the bacterial symbiont in this system. And so the first thing that we did is we did RNA-seq analysis across a pregnancy cycle. We did non-pregnant females, early in lactation, pre-lactation when they when there's a embryo in there but they're not producing milk yet, early lactation, late lactation, and compared to males. The main thing that you see is up here in the upper right is during the pregnancy cycle, you see a massive increase in cytoplasmic translation and ribosomal large subunit biosynthesis. This is to produce a lot of proteins, probably to produce the milk. We're able to characterize here on the right here some of the most important proteins associated with the mechanism live birth. A lot of them are highly expressed in the brood sac, and these represent most of the milk proteins. So similar to the Glossina, they have milk proteins that represent almost all of the highly expressed proteins in the brood sac. So this allowed us to actually kind of overall understand the molecular mechanisms of viviparity in Diploptera. We also did functional knockout, some juvenile hormone genes, a few other things to establish mechanisms of this. Overall, this gave us a good idea of what is happening during Diploptera reproduction. So along with understanding the actually aspects involved in milk production and live birth in Diploptera, we wanted to see is, all right, what are the microbiome associated with this cockroach? So most cockroach species, they have a symbiotic bladobacterium that occurs in there. And so when we did microbiome characterization throughout the pregnancy cycle and early in development, what we see is that embryos and neonate pretty much have only bladobacteria within them. So it's mainly that this protein is probably very critical to these stages during development. There's no other microbiome. As first instar, after their cuticle has melanized and they begin to move around and start eating and ingesting water and that sort of stuff, their microbiome begins to kind of move up to what it will be throughout the rest of their life. And so this is more diverse and probably represents all the bacterial species present within the gut, along with bladobacteria. Even in these, even after first insars, the bladobacteria represents a huge amount of the bacteria associated present within these cockroach species. So to figure out the potential role for this bladobacterium during reproduction, we actually said, all right, let's sequence a genome. So we actually extracted the fat body from this cockroach and then did enough DNA sequencing at a depth to actually assemble the genome. So we assembled the bladobacterium genome for Diploptera punctata, and this is the overall characteristics of this, and with the goal of figuring out what it's potentially doing for this cockroach. In multiple other cockroach species, bladobacterium has been associated with amino acid metabolism. And so we assumed that it would have a similar effect here. And so what we were able to actually show is that it actually can synthesize most of the proteins that are necessary, particularly the essential amino acids. And what the main ones that could be done, and particularly in the metallothionein synthetic pathway, similar to other cockroach species, is fully lacking, but synthesis can occur in conjunction with the host, and this will be critical for juvenile hormone biosynthesis. And levels of juvenile hormone synthesis are critical for regulating the development of this developing embryo within the brood sac. So overall, it's probably undergoing interactions with the host in order to properly synthesize methionine. So along with these recent studies, we actually just not too long ago finished sequencing the genome for this cockroach. We're really interested in it as kind of comparable to termites and other cockroach species, but also for what is unique about it that allows it to undergo live birth. And so here's some overall stats on it. It's a high quality genome. Overall, it has a high BUSCO value. It has a fairly low number of contigs, and that are overall very low. So it's not chromosome level, but overall it's a very high quality genome, and we're currently working on the final analysis of this, and probably should have this paper out within the year. So overall, I would like to thank a few people for this during this presentation. Dave Billinger, Rick Lee, mainly they allowed me to go to Antarctica to do the work. JD Gantz, who is right here in this gray jacket, he helped collect a lot of the Antarctic samples, the polymerization for the Antarctic samples, the NSF for funding both the Polar Project and the Drosophila Project. Funding was provided by the University of Cincinnati and Miami University, and the USDA, and then the Children's Hospital, and for different areas is there for different parts of the project overall. I would think multiple other for their individual projects, but I think them within their individual sections, such as Mike Pollock, Cassie Hooker, Francis, Jorge, Jeff, Carly, and all the contribute, Emily, and everybody else. So, but mainly overall, they are very essential to actually finishing up these projects. And so just to kind of leave it here, I've been pretty lucky in my research throughout my career. This is actually one of the sunsets from Antarctica when I was there the last time. You would go up, take a picture, and say, oh, that's a beautiful sunset, and then go up again, take another picture, it's a beautiful sunset. And they would just get prettier and prettier each time. And then you would say, oh, that'll be the best one I'll see the entire time down there. But you get very lucky down there, and the staff's great. They allow you to do all the research and focus on the time. So, and the other thing is I'm usually always recruiting new graduate students and postdocs. And even if it's not me within the lab, I have a lot of great colleagues who work on insect systems. And so if you're interested in the University of Cincinnati or the area and would like to talk to me about being a grad student for the department or a postdoc, I would be happy to answer any questions about that if you just shoot me an email. All right, on that note, that's my presentation. I was trying to keep it a little bit shorter and focus on these 3 topic areas. I hope overall you guys enjoyed it. If you have any questions about any of the specific areas, feel free to shoot me an email. Most of the papers are currently out now. I believe one of them, we're working on the Diploptera genome, and then one of the other Belgica reproductive papers was just recently published and posted on bioRxiv. So if you have any other questions, shoot me an email. I believe we'll have a question to answer period at the end of the presentation. So thank you for taking the time out of your day to listen to my seminar. I know everybody's swamped now, but I would like to emphasize everybody, even as like faculty, and everybody seems to be a little stressed out now. And so if you feel like you're getting stressed out and you need to talk to somebody about mental health or those sort of things, make sure you do that, because it's a pretty stressful time, but everything should be getting better with everybody getting now vaccines, cases are going down. So hopefully in the next year, life will return a little bit more to normalcy. So thank you, and enjoy either the next presentation, or I'll be on in 1 second to answer some questions. So thank you.
All right, thank you so much, Josh. Another very interesting talk. Before we move into the discussion, I thought I would give you a chance to answer a couple questions live here. There's one question from Ellen Lee I thought was particularly interesting. It says, regarding the protective gel, how does this gel compare in composition and function to protective jelly layer surrounding amphibian or fish eggs?
You can hear me, right?
Yes.
Okay. Functionally, it's probably very similar, but the protein content. So there's a lot of proteins in there. They probably have a lot of like glycosylation, phosphorylation, those aspects. But there is some significant differences between them. But they probably prevent egg dehydration, act as an early nutrient source, and that sort of thing. So they probably have a lot of similar functions, but overall, some of the proteins present in there are pretty different.
Okay. Okay, and there's another question from Rob Waterhouse about milk proteins across different insects being homologous. Can you address that?
So yeah, I typed in the answer for that one, but I'll go ahead and answer it. So I would say they're functionally similar. And so kind of similar to what I just said with the frog egg gel and those aspects. So in Diploptera and Glossina, like there's a lipocalin in both of them, but they're probably not from the same common ancestral gene that drive them, but they have a similar functional aspect of probably having to carry some small hydrophobic, like moiety, so it's like a little hydrocarbon or something from the mom to the developing larva, or a vitamin or something along those lines. And then they both have their own little groups of proteins that are highly expressed. But then the relationship between those is probably very similar to like the relationship between mammalian or marsupial milk. So they have to get the same thing of getting a lot of nutrients to this developing offspring pretty quickly. So they're highly packed, nutrient rich lipocalins and other small proteins that can provide this source to allow this development of this either smaller juvenile or intrauterine individual or interbrood stack.
Okay. Okay, and then I had a quick, I guess it was a question. That video of the fly giving live birth, I think probably intrigued a lot of us, maybe horrified us a little bit. Is there an extra fitness cost associated with a female having to give birth to such a large larvae?
I mean,
compared to
yes,
just laying an egg mass?
Well, yes and no, not as much as you would think. When they lay the large egg mass, they're usually laying a lot more eggs. So the overall like nutritional investment is probably still higher, probably still significantly higher. But it's kind of these ones, is it an R strategist, K strategist? Like you can lay a lot of eggs, but then maybe half of them get eaten by, if for example, a mosquito, an aquatic beetle, or something along those lines, versus tsetse flies who kind of lean towards the one individual offspring and provide a lot of nutrients. They deposit that and immediately pupariates. So there's probably a higher number of survival between one versus the other. So it's just kind of whole benefit trade-off aspects. So there's some ideas, and we have some cockroach data to kind of support it, that under kind of periods of like environment instability, like the live birth aspects may be a little bit better, particularly like if you can't find water sources. If you're a little tiny larva, you'll dry up and die really fast, versus the mom may be able to fly around, find a walking bag of blood and water, which would be us or a cow, and be able to find that sort of stuff a little bit easier. So there's kind of, you got to think about like the cost benefits of that. And that's pretty much how I think about it.
Okay, cool. Thank you. I'd like to thank all our speakers again too. I know some folks may not be able to stay for the discussion part. So thank you everyone for your participation and putting together such great talks. I think now we'll move into the more discussion. So if all speakers want to come up live here, you can always, interrupt me if you saw a question that was really interesting for your system. Feel free to jump in at any point. I was just going to tackle a couple of these. There was a small thread of questions that were all kind of related to chromosome size, Heath. I don't know if you want to address that. Some people asked maybe if the dot chromosome fused more often because it was small. And then I think somebody else asked, well, after they're fused and they become quite large, are they less stable at that point? So if you could address those questions.
Yeah, so first off, the model that we have right now doesn't address chromosome size in any way. And actually, we kind of went around and around with reviewers around Christmas time, trying to decide whether or not our model was complex enough. Certainly, the size of the chromosome seems like it should have an impact on things. Our model doesn't include that. And somebody asked, like, what would you add to this model? And that's one of the things that we're working on adding to it, is because it would make sense to be able to put that in there in groups that are achiasmatic. I think I showed how it could have an impact, but I also think it would have an impact on whether it can fix. But I think it could have a big impact on how often that type of mutation, a fusion between a large chromosome, involving a large chromosome versus a small chromosome, occur. And so I think that would be a great thing to add into the model. And we're working on that in an analysis across all of Diptera right now.
Very cool. Very nice. Oh, sorry. Someone asked about the Slack channel. I should probably address that before we get too much further. So in the chat, there was a direct link if anyone else is curious about Slack information. Okay, sorry, I just wanted to throw that in there. So while we're talking, Heath, do you expect reproductive isolation between Neo-X carriers and wild type individuals in the population?
So when we look across empirical data sets, we see that in some specific cases, there is reproductive isolation due to a single fusion event. But also there are many examples in empirical systems where if you have a single fusion event and no other rearrangements, that it doesn't impact the ability to properly segregate chromosomes. It seems as though in most cases it takes a fusion plus some other changes too. There could be inversions or translocations. And then once you have multiple changes, then that's when you really start seeing an impact of different karyotype morphology.
Okay, thank you. Let's see, Amanda, I believe several people commented on the BUSCO score and why maybe you couldn't get over that 90% single copy ortholog number. Would you mind addressing those questions?
The answer is I don't know. So all of these assemblies, Anna pointed out that I'm just reporting the complete single copy orthologs, so not the duplicated rate. If you include that, we're over 90%, but that's not the majority of the missing BUSCOs here. It's in the missing category. So I don't know. We need to look into that, and I'm very open to ideas and how to tease that apart.
Okay, yeah, I know a lot of insects have a lot of repetitive segments, so maybe things just aren't aligning correctly, or maybe that's a potential, I don't know.
Yeah, I mentioned the repeat content could be a possibility, and I think that is probably true, at least in part, in the hybridizing species because they have a much larger genome, but yeah.
Okay, all right, great, thanks. We have some new questions coming in too. Heath, I think this one's for you. Why do some groups have large numbers of small chromosomes while others have small numbers of large chromosomes?
I mean, that's like the question that I want to answer eventually. I want to know why we see the distributions that we do. Because even in Lepidoptera, there are some species that have a crazy number of tiny chromosomes, but there are also species that have far fewer chromosomes. And so what can explain this distribution? What are the forces that push species towards one extreme or the other? I don't think that we've done enough analyses kind of across the tree of life to really say what's going on. We have tested in Hymenoptera, there was this very clear hypothesis put forward in the 70s that eusocial species should have more chromosomes because it allows them to produce more genotypes in their offspring. And so if you have this huge colony, you want to have lots of different genotypes. So a parasite or a bacteria couldn't wipe out the entire colony. And so we collected all the Hymenoptera data that was available out there, and we found that actually eusocial insects don't have a higher chromosome number than solitary Hymenoptera. And so I think that I think it's a question that we can't answer yet.
Okay, yeah, some complicated, big questions there. Let's see, I'm just gonna bounce around questions. So y'all pipe up if there is something in particular you wanna talk about. Since this is Arthropod genome section, I think I'll highlight this question to Josh. How big are cockroach genomes, and are they easy to assemble? Are they one of those highly repetitive, hard ones?
I mean, so Mark Harrison and Eric did a lot of the assembly for this one. We had good depth pack biosequencing, and it's not chromosome levels. Like if we got some high C information to go along with it, we could probably quickly get into chromosomes. It depends on what we want to do with it. It comes down to as long as you get enough, and it's more or less material from highly inbred males that so it's good information. We had enough depth of sequencing to get a really good assembly, and so it wasn't any more difficult than the other ones. And because it's such a big assembly, like in comparison to some of the early cockroaches that were only Illumina based, it's a really good assembly. And really, it just comes down to those aspects of getting enough high quality like pack biosequencing, and then adding on like some sort of like which we didn't do, some sort of chromosomal kind of scaffolding to do, and then you get a good one. I think it's like 2.5 to 3 gigs, and so they're big assemblies, but if you get enough data, they'll assemble well.
Okay, great. Great. It seems like early on, Amanda, you had a very involved question that was quite long. Would you like to walk us through that one? See, David.
Yeah, so David was asking whether the hybrids could be optimized for this new fitness landscape in the novel environment. And I think that is motivating a lot of our work, this hypothesis. And I wrote here that we do have some evidence for that with the Carinata subleniata hybrids increasing in frequency, especially in that time series data set. But disentangling this from demography, like the release size and how those release sizes overlap, is really, really challenging. And the data about those releases is incomplete. So it's a good question. We're working on tugging at that, but it's an open question.
Okay. All right. Thank you. Speakers, was there anything you would like to address? before I just start randomly picking some questions.
So I wanted, if I could, I wanted to slightly embarrass somebody else on here who she's probably running the AGS 2021 thing. So I'm going to share my screen, which I think I can still. So I just wanted to share this very quickly that Pia Olofsson's Stablefly genome paper will be coming out actually tomorrow. So I just wanted to give her congratulations on that. And that's been a lot of work over the last few years. And so I just wanted to share that, and everybody can see that correctly.
Yes, yes. Thank you for sharing that. For those of you who don't know, Pia is the one that has been working the videos for this symposium. So yeah, that's excellent work. Good job. Good job. Thank you for pointing that out. All right, well, we have about 15 minutes left. I don't know if anyone has any burning questions they want to put in the chat. I just want to kind of wrap things up. We had 3 very diverse talks today. They were all wonderful talks and gave us a lot to think about. I think it always surprises me how diverse insects are, and how like there's so many different things to study, and they're all interesting, and they all kind of are encompassed by this evolution and diversity of insects. And I think it's really a fun topic to talk about. And so I appreciate all 3 of my speakers today. I know if you could hear everyone in attendance, they'd be clapping. These were fantastic talks. And I also want to thank the organizers. So kind of behind the scenes, we had a lot of ARS folks, Brad Coates, Glenn Hayes, who did a lot of the IT stuff, Pia, who we just gave an awesome shout out to. And then also I appreciate I5K and the AGS group for allowing us to use all their tools, their Slack channel, their YouTube channels, and all their means to promote this symposium. So everyone out there that is still listening, remember, if you are listening to this as a recording and you have a burning question, you can go to that Slack channel. Speakers will check periodically to see if there's some new questions. And then also there's 2 great sessions left in this symposium. And so all that information is found on the I5K AGSX website. So you can get links to register if you want to share with colleagues. They can register there, get information about Slack, and get information about the next 2 sessions. So I want to just, again, thank you everyone for your participation. And if there's no other comments from the group, we can move this to the Slack channel.
While we're still here, can I answer one more question instead of typing it?
Of course.
Somebody asked about what I had mentioned about Hymenoptera and whether I think eusocial Hymenoptera don't have a higher chromosome number because they have higher plasticity of expressing genes with epigenetic mechanisms. I was just going to say that my collaborator on the Hymenoptera work is Laura Ross at Edinburgh, and she did a really nice analysis in our study of Hymenoptera that suggests that actually it's other mechanisms that Hymenoptera have evolved to generate greater genetic diversity in a colony, with some females mating with multiple males before they found a colony, and some species having multiple queens in a single colony. And both of those would kind of remove the pressure that you would expect to lead to higher chromosome number. We didn't look at the thing that they suggested in the question, but those 2 things did seem to have an impact.
Okay. Okay, great. Yes. Was there any other due questions that I missed?
No, I just have a parting comment on Lindsay. You forgot to recognize yourself for all the work that you did to make this possible, as well as Glenn Haynes, our master in the background, who does all the IT and makes the Zoom links, sends out all the reminders. It's been a great help on this and multiple other things.
Yes. Thank you, Brad. Okay, everyone. Then with that, I will, there's Glenn. I will see you all on the Slack channel, hopefully. And in parting, I'm going to go ahead and share that info slide. So if you want to get the link for our website, you'll be able to do that. All right. So, well, It doesn't want to change. It did this to me earlier. But anyway, without any further ado, we will see you all in your questions on Slack. Thank you very much for attending.